Good afternoon, good morning, or good evening, everybody. My pleasure to welcome you to our webinar about connecting text analytics and multi-omics sequence analysis in order to gain further insights into drug discovery. I'd like to introduce you to Dirk van Hyfte and Sébastien. Dirk van Hyfte, who's the CTO of BioStrand, and Sébastien, who's the data scientist at our company. Who will, between the both of them, further introduce you to our take on NLP technology and how it is implemented and can be implemented into your workflow, guiding you through some practical examples. On the agenda, well, we have our take on the fundamentals that we deem to be very important in order to provide very good natural language processing. So we'll talk about how we can integrate all these diverse knowledge domains while still gaining and remaining on top of the different entities that are associated to it. Again, also that's important because, well, what our philosophy is in this case is that you need a semantically driven approach, because this is crucial in the domain of life sciences and drug discovery. Next to some technological introductions, stick around because Sébastien, in the second phase of this webinar, will go over some practical applications and implementations of how you can exploit our NLP technology and how it functions into notebooks. So on that note, a very warm welcome again, and I'd like to pass the word to Dirk van Hyfte, who will guide you to the first part about our approach and take on natural language processing.
So thanks a lot, Arnout. Well, as an introduction, indeed, at BioStrand, our mission is to build the next generation technology that makes omics data analysis truly effective. And what is our global view on drug discovery is really how can we go from the sequence or the different sequences to insight. And here we combine a unique way to not only start from a data-driven approach, so really from the bottom up, from the data itself, but also combine that with prior knowledge and how we can combine that. And if we have a broad perspective on our platform, then you can see that we support 4 different quadrants. If I start on the left side, on top, there you see that we have the whole sequence omics integration. So we have a way to integrate DNA, RNA, and amino acids, both in public data sources, but also in your own proprietary data. And this sequence, so call it the syntactical information at the sequence level, is also combined with structural information, and even further with unstructured textual information. And especially in this life science and healthcare space, there's quite a lot of information that's really valuable for drug discovery, and that resides in, for instance, PubMed abstracts, scientific papers, electronic patient records, and so on. And so especially today, we will focus in on how we can extract all that knowledge out of that unstructured information. And then the 4th quadrant is how you integrate it with really formal knowledge that is also in knowledge bases or data banks. So the first challenge is indeed how can we extract information from many data sources? And that's really a challenge. That's the difficulty. And so especially in this omics area, where you have data at the DNA, RNA, and amino acids in different data sources. And so this challenge is how can we integrate all these sources together? And the second part is also that we have to deal with quite a lot of unstructured information, both at the sequence level, but also at the scientific knowledge. That is, most of the scientific knowledge is in unstructured format, just in text. And what you typically see that researchers that are working in this field, they depend on bioinformaticians to integrate all these data sources. And so we at BioStrand, we have built already our first platform where we can integrate all the data from different data sources at the three omics layers. And so we have an integrated way so that you can look immediately, starting from a sequence, what is going on at the DNA and the RNA level. And you see that on the right part of our screen, where you can see your platform in action. So you can start off from a sequence and then immediately look where the same sequence or similar sequences are in different data sources. And also important that you can see quite a lot of metadata that is associated with this sequence. And also in our platform, you can really slice and dice that data. So it's based on a multi-dimensional model where you can further filter down to the specific information you really need to have. And so our whole platform is based on a twofold indexing principle. On the one hand, we have our Hyfte patterns that helps to index these three different omics layers. And on the other hand, we also have our semantic entities or top concepts to glue together also the NLP word, so the unstructured textual information. Next, let's say, challenge is how can we bridge all the gaps? And that is typically what researchers are faced with. And so how can we bring the experiment or the data that is generated by the different experiments, and how we can bring that in connection with the knowledge that is, for instance, resides in literature. And that means we need to have a different view, a different mindset, even to look at the data. So the first challenge is indeed how we can turn unstructured information to structured information. And so at the sequence level, for instance, with all the raw reads, that is fully unstructured information, just a combination of letters. And there the challenge is how can we map that to, for instance, known references like the human genome reference and to look for certain variants. On the other hand, you have the scientific literature, so the scientific knowledge, that's also unstructured information. And there also we need to look for ways how we can turn that in structured information. So because from once you have structured data, then you can really apply computer algorithms to make sense out of it. So we will present our natural language processing technique where we have a unique way where we combine different approaches and using also AI so that the machine, the computer can start reading text and detect right concepts, so meaningful word groups. Let's have a look on how we do the first step in our natural language process. And here I start off with a sentence: 2 patients are suffering from congestive heart failure. So the first step we do in our process is what we have called smart indexing. And that smart indexing is based on the principle that we first look at the linguistic representation of relations. And mostly they are verbs, not always, depending on the context. And in this example, that relation is are suffering from. And once you have detected this relational element, then you can say that everything that's left or right from that relational element that are referring to concepts. And in this way, we understand that the sentence means that it's going about 2 patients that have congestive heart failure. So we are able to detect this multi-word groups and also define that semantic word boundaries without predefined knowledge. And it's really important to stress this. So without knowing anything from a certain domain, we are able to extract that information. So it's not tuned to a certain specific area in life science or in healthcare. No, it's a very generic way to do that. And here I also further will look into how it's so important to have to grasp the right meaning of these meaningful word groups. And so let's start off with an example that we look at the word dopamine. And dopamine is indeed a small molecule, neurotransmitter. But when I refer, for instance, to dopamine receptor, that means something totally different. That's really a drug target. So it's a receptor, for instance, in the brain. Well, on the other hand, when I refer to dopamine receptor antagonist, that's a drug. That's a drug that can interact at the dopamine receptor. But you also have word groups like dopamine receptor gene, that's a small molecular sequence, or dopamine receptor gene mutation, that's a physical process. So you see that, especially in the area of life science and healthcare, where we try to understand the relation between certain drugs and receptors, it's really important to grasp this complex, meaningful word groups and to have a good understanding of it. And so the first step in trying to give meaning to these word groups is by using dictionaries or ontologies. So that's really the top-down approach. So predefined, you do a mapping with knowledge that already exists in a dictionary. And let's suppose that you have in your dictionary only the word dopamine. Then in all the three different cases, it will be mapped to just dopamine and ignoring the more specific semantic meaning of the word group. And that's an important part, because if you already start making mistakes in having the right semantic labeling of your word groups, then later on in your process, once you want to start looking for certain relations, then it becomes problematic. So the solution is that only with working with dictionaries or ontologies, that's not the right way to go further. And so that's also why we have looked at the new techniques like transformers and especially like BioBERT and that kind of AI machinery, and see how well they are doing in this capturing the semantic meaning. So for instance, here you have a sentence, PML binds, and so on. So it's a rather complicated sentence. And so the way we analyze this sentence is by first detecting this concept relation concept patterns. And again, without predefined knowledge, in a very generic way. So it's not tuned to a certain domain, but in a generic way, we can capture this type of sentences. And as you see, we are able to find this concept relation concept patterns. So for instance, PML binds is the relation, MK2 is a second concept, and so on. But also at the end of the sentence, you see P338 MK2 interaction, that is the full concept. So again, we are able to define the semantic word boundaries. And then if you look, when you use, for instance, this transformer architecture to do the mapping like BioBERT, what you see then that only small parts of this sentence, or even at the concept level, are mapped to, for instance, a certain gene in, like you see with this PML1 in the beginning of the sentence. But it's also important to see that this BioBERT even does not recognize the whole full concept like P38 MK2 interaction, because it's really split in different parts, and even the word interaction is not mapped at all. And we call this part that is not mapped a leftover. And so this is a part of the concept of the multi-word group that is not tagged yet by this BioBERT system. And so we don't have any meaning of that part. And we also looked further into that area to see, okay, what are the challenges when you just use, for instance, this type of transformers. And so we did some analysis on around 300,000 PubMed abstracts, and then use different transformer models and see again how well they map or they find certain concept classes like disease, drug, genes, and so on. And just a good way to read this graph, on the y-axis, you have the leftover ratio. So let's suppose if we look at the class disease, then you have all the word groups that contain the word that point to a certain disease. And in blue, so on the left side of that specific word group that points to a disease, we have some leftovers. So parts of that concept that are not tagged. But also on the right side, and that's in orange, you see that a part of that concept is not tagged. It's also important to understand that, especially in English, the right part of a concept is really pointing to the specific meaning. Again, refer to that example, dopamine receptor or dopamine receptor antagonist. So that last part, antagonist, is really defining that it's, in this case, going about the drug. So having a good mapping of the full multi-word group is really important. And what you see that if you count, so the left and the orange part, then you have around 30, even 35%, depending on which class you are looking at here in this graph, but around 30 to 40% of the multi-word groups are not fully tagged. So that means you are losing information, you don't have the right semantic meaning of it. And that has its consequences when you in next steps try to extract relations and try to understand the meaning of certain interactions. And especially, I've highlighted here with these three circles, so in the group of diseases, drug, and genes, well, this is rather problematic. So you need to solve that part. Here is another example, looking back to word groups that contains the word dopamine. For instance, you have central dopamine or mesolimbic dopamine. So that's one word group. On the other hand, you have dopamine agonist or dopamine antagonist and so on. So that are different word groups. And what you see that this BioBERT model, it all assign this to the same semantic class. While, again, as we have seen before, the red part like agonist, antagonist, and so on, that's really referring to drug and drug classes, while dopamine turnover, dopamine synthesis, and so on, that's really referring to another semantic class, namely processes. So again, it's really important to assign everything to the right semantic class, and it seems that these classical transformers are not doing that. So we have a unique combination of, in the first step, do this smart indexing. So again, without predefined knowledge, identifying these semantic word boundaries and to define concept relation, concept patterns, and then map that together with ontologies and dictionaries in combination with these transformers models to assign the right semantic meaning of this concept. And that's the best way to indeed start doing relation extraction and trying to identify knowledge that you can extract out of scientific papers. So if you step back and look, okay, why are we so unique? Well, we have a bottom-up approach on the one hand, at the sequence level, where we do the integration with our Hyfte patterns and can easily look at the different omics layers, again, at the sequence level. On the other hand, we have our very specific NLP technology or doing our text analytics, and that is combined in one single multidimensional model. So if we look back, to the way we have structured our platform with not only the sequence integration, but also the unstructured textual information. And that's exactly what Sébastien will further discuss, how we make the bridge between the sequences and the unstructured information, and that based on the Hyfte patterns that are a kind of gluing point of all this data.
Okay, all right, so I will take it from here. So to bring a little bit context into Hyfte patterns is a technology that basically encompass a data structure that we found in both, in all AA sequence, amino acid sequence, RNA sequence, and DNA sequence. And these different Hyfte basically connect all the different omics layer as a new transversal language and encompass different information on function, position, and structure. And so we basically use that as the central, the core of the indexing solution. So do I have control? Yeah, okay. So basically, in order to understand a little bit more how this Hyfte are leveraged in order to make all these connections, let's first take an example from two sequences. So these sequences contain some Hyfte, and these sequences contain some Hyfte in common. In this case, these are if2 and if4. And associated to this sequence, you can have different amount of annotation, like gene ontology annotation, structural annotation from CAT, etc., etc. And there are also unstructured metadata, for example, the sequence descriptions that you can find in a FASTA header, for example. And what we identify as structured data are all the data from which you have a database that actually put all this data in relationship with each other. And the textual metadata, that's still quite valuable if we want to extract information of it. And by using the BioStrand NLP technology, we can extract from this textual metadata top concept. And we can do that for any sequence that we ingest in our system. And once we start looking at these top concepts, we can actually relate all of them through the Hyfte patterns according to their sequence. And so we can perform a twofold indexing approach. We can either index it based on the Hyfte pattern that we find, or based on the top concept that we retrieve from sequence metadata. And so this technology is already implemented in Retrieve and Relate, because in Retrieve and Relate, we can start from a sequence as a query, for which we can find some homologous sequence, but we can also do a text-based search where you simply add some description, some vague description of what you want to find, depending on how precise you want it to be. And you get back, relevant information by using these concepts that have been extracted from them. And so what is important to remember is that in Retrieve and Relate, we have done this process for more than 40 million sequences. And that's from several public databases like GenBank, Tremble, UniProt. And it's also possible to perform this ingestion on private data that you can upload. And so to brings the relation of the sequence data with textual data, we basically made an NLP ingestion pipeline that basically gets the text and parse it with the BioStrand technology. And this actually leads to what we call CRC triplets, which are direct relations that we can find in text, but also a sentence, alinea, or full document that contain different matrix of proximities between the concept that we retrieve in this document. And once we have done that, we can basically, through the concept that we retrieve in both the sequence metadata and the scientific literature, combine and link more than these 400 million sequence with 33 million document. And one thing that we have developed for now is basically a graphical interface that allows to well, do some treatment of this ingested data, like reviewing what we have ingested. So in this case, we started from PubMed abstract as a starting point in order to do that. These abstracts have been mapped to different dictionaries, and from the concept that we are retrieving, we can perform some set analysis. This allows you to define specific subdomains of PubMed. For example, if you want to only look at things concerning with cancer, then you can simply combine different concepts related to that in order to build a subset. And also, it is also possible to explore all of this data and to derive different metrics like frequency, TF, IDF, time frequency, inverse document frequency. And if you want to have more information on what we have done, we already discussed about all of this at the previous Biostrom webinar from last November. And so, in order to understand a little bit more the use case that I will describe in the following slide, we need to understand a little bit the flow of how we proceed to do things. Basically, if we start from text, and from raw text, we can extract meaningful concepts based on these CRC triplets. These concepts can be tagged individually with different kind of type, depending on the technology that we want to use. We can use dictionary matching like UMLS. We can also use transformer-based technology like BioBERT. We can perform also normalization of concepts that actually relate the concepts that are retrieved from text to structured data on different database. And once we have all of this information, we can start extracting the information to build graph that represent in Knowledge no longer as text, but in a graph format. And in the following, we will identify different graph, one that we call proximity graph, which are graph that display abstract relationship between concept, depending on proximity, semantic proximity in a text, and also direct relationship graph, which are basically direct relation between different concepts in the same format as what we have shown previously, based on the triples. So concept, relations, concept, etc. And basically, using that graph, you can go deeper and deeper, finding new relation, new concept, and so on until you can define your hypothesis further. So now let's have a look at some example of the small use case treated with Python. So the very first use case you are interested in is how we can query the ingest text data with Python. So in order to do that, we built a very simple Python module that is the API from which we can perform different kind of queries that can be integrated into any kind of workflow. And what is important is that we keep the information from the original sources. Like, well, in the case of PubMed abstract, we keep track of the PubMed IDs and so on, and we can always trace back whatever we find as concept back to their original source. And amongst what we can extract, of course, this concept relation, concept triple. We can extract as also what we call path, which are basically sequence of entities. So once you remove elements that are not of height, well, that do not hold further information, typically a stop word, etc. We can also extract the source and the entities metadata, like, for example, named entity recognized through a transformers-based model, normalized entities, dictionary matches, ontologies, and so on. So all of this is stored somewhere in our database, and we can query with these different the different functions from this module. So in this example here, what I am showing basically is that I import a client. The role of this Python client is to interact with the database and to perform different queries. We have the knowledge that is stored in different domains. In this case, I will simply call the domain PubMed because that's basically all of PubMed's abstract. And we can perform a query by submitting a query. In this case, we basically have a single keyword forgot in it, and we want to extract all the CRCs from this query on the domain that we have defined, and we limit that to only 20 of them. So this is our structural parameter that we can say, and we store that query ID somewhere until it gets processed on the server. Once it's processed on the server, we can get back, well, we can check the query status, and if it's fine, if it's processed, then we can get the results back. And here we are dumping this result in JSON format. So basically, here we add one query ID that is this number, and because I asked for concept, relation, concept, or CRCs, as we call them, we basically get a list of CRCs given into these dictionaries where we have relationship between what you have on the left and what you have on the right, as well as the frequency and possible matches from dictionaries, recognized entities, etc., etc. In this case, because I did not specify to retrieve any matches, it's not displayed, but this is something that is a possibility. So this is one example amongst many different queries that we can make and build some application around this. Then, once we have ingested the text as a data, and once we have this Python module to perform basic query, then we built a simple application around that basically transforms the data that we have stored in our database within relational table into a graph structure. So currently, we identify two kinds of graph, as I said. So we have this abstract proximity graph and the semantic graph. And basically, if we want to perform a query, we can follow this workflow. So first, we need to import the different classes that allows us to perform these queries, as well as some element of configuration. Then we define a query. Basically, it's a list of concepts that we want to query in this case. There are some parameters that control the size of the graph that we get back, or informative it will be, etc., as well as different optional keywords. In this case, I specified a new domain on which I want to find my query, and I do not want to have any metadata with respect to this. And so once I instantiate my class, I can perform the query. First, some optional pre-processing steps. So basically, I feed to my class a list of stop words, as well for concept as for relationship, because we treat them at a different level. And I also store here, basically, I ask the program to get some pre-computed TF-IDF metrics that I will attribute to different nodes. These different steps are optional in this case. Then we basically perform the query. So the query is done through a three-stage query that basically defines the scope of what we will get back as a result. Once this is done, we can basically make a graph based on the result that we got back. On top of this graph, we can also do some further processing, but this is optional, like we can perform name entity recognition on top of the graph, as well as to strip the graph that allows to remove information that are not directly related to the element that we have set in the query. And that's a way basically to remove information if the graph is too cluttered. Then we can save the graph. Here, I have decided to save it into a JFX format so that it can be open with open source program. And I have done that in this case with Hyfte. And so here you can see we have the graph that has been generated from this particular query, where we see abstract relationship between different elements like SARS-CoV-2 and COVID, as we can see here at the center of the graph, and different entities as well, like in this case, Mycobacterium tuberculosis, and so on. So that's basically what we have here. All the ages that are shown on this graph have a feature that is not displayed for clarity, but is basically a semantic proximity that quantify how close two concepts are with each other, with respect to each other. And in this case, the node also carry different potential metadata, like, for example, the PageRank metric that is computed on top of the graph, Louvain partitions computed on top of the graph, potential annotation, like those that are extracted from named entities, and so on and so on. So this is an example of proximity graph. And we can also extract another kind of graph with a very similar kind of query. This time we're using the semantic graph class. It's the same workflow, basically, some pre-processing step, a query, and we basically make the graph and save the graph. And this time we want a semantic graph. Semantics basically means that the ages have a semantic meaning. There is an action or an actual relations that is well defined between different entities. And here, this is an example of graph that we can have from this. So basically, the node in this case has the same properties as in the previous graph, but this time the ages contains new properties, like, for example, okay, what's the relationship between these? So we have, for example, that coronavirus disease 2019 is caused by severe acute respiratory syndrome coronavirus 2. Okay, so this is just a small example of what we can get back. But the most important aspect to remember in this case is that we can basically have these relationships and to find, for example, causal relationships between entities and so on, and to find also proximity in a greater scope between different entities, which can have its purpose, for example, for drug repurposing. So now I've shown here these two small application. There is also another thing that we have developed at BioStrand is how can we get from sequence information to graph. So as I've said previously, we have indexed the sequence metadata with the NLP technology. And so it basically means that at the multi-omic analysis level, we can have a set of sequence that you can extract from experiment or biobanks whatsoever. And this sequence can have relationship with each other. For example, you can have relation between epitopes or paratopes. It does not really matter what's the exact nature of the relation in this case. It simply means that, okay, we'll use that as a baseline to perform a query. And what we do once we have all of these sequence, we need to retrieve the annotations to normalize these annotations, eventually to filter them based on your own interest. And then once we have that, we basically go beyond the landscape of sequence data, and we go into the landscape of textual data that has been ingested. Basically, the top concept that we retrieve, because they are well targeted, we can use on-target lookups on the text that we have ingested. And so we can associate sequence concept with literature concept, find intrinsic relationships between these concepts, and on top of that, we can put some ontologies, dictionaries, labels. And from all of this, you can basically extract knowledge that you could not extract in a straightforward manner just by reading the text or just by processing the text in, let's say, if you do it manually, that's gonna get much harder. And so you basically have a very high level view of what you have in your data, and with relationships that are meaningful and that can be used, for example, to perform any downstream task with a computer or an algorithm, etc. And so let's have an example, a very small example on what we can do with that. So here in this case, I have taken a small snip from the SARS-CoV-2 spike glycoprotein. So we have heard a lot about it in the news. Not all of us actually are working specifically on that, but yeah, I guess it's ringing bells to a lot of people. And if we copy paste the snip into retrieve and relate, we find back a lot of sequences that are similar to this one, a lot of representative sequence from different databases. What we see in this small example is that we got back mostly PDB sequence that contains a PDB model associated to them. And some of these PDB sequences have different annotations. So here, most of them are the same, but you can see that, for example, at some point you have SARS-CoV-2, which explicitly appear in this case. What we have done is that basically all the sequence metadata is annotated as such. So we will perform a lookup inside the full database in this case. And so this is what we do. So basically we take, in order to perform this query with Python, we use here NLP index client, as well as a function that is called get top concept. And we use also something else that is the Hyfte parser, which is a parser that allows us to retrieve Hyfte from sequence. And so we initiate the clients to get a direct connection to the index. Then what we do is that we basically take the sequence we are interested in. We get the Hyfte from this sequence. This is what this line is doing. Then once we have the Hyfte, we can basically use these Hyfte to get back the top concept from the index. And here we have a sample of what we got back from the index. So we see quite a lot of spike glycoprotein. Sometimes we can even see some other concept concatenated to spike glycoprotein like fibritin. This is due to these fibritin models that are happen to the spike glycoprotein in the PDB models. And there are also different candidates that also appear here. It's not always, well, here, is it always SARS-CoV-2? It's not always mentioned, but yeah, you have different occurrences here, and these occurrences are given here by the count. And once we have the stop concept, we can basically use these as to construct a query from which we basically reuse the previous code from which I've made this small example of graph. So we basically defined the query. Then we define the query body that contains different parameters that expand the size of the graph, depending on what we are looking for. And well, okay, here I have made these optional pre-processing steps, performing the query. Then I get back a graph from this query. I have here quite some nodes and some edges as well. And what I have done is that I have basically stripped the graph only to keep some relationship between the element that have been used initially to define my query. And I save the graph into a file that I can open with Gephi in this case. So here we have an example of such a graph where I highlight here spike glycoprotein, and we can see directly the relationships that we have between different entities. Like we have this SARS-CoV-2 that's related to the ACE2 protein. And well, we see ACE2 appearing quite a lot as well. That's because it's been heavily mentioned in the literature. It's the whole mechanism that actually make us vulnerable to the SARS-CoV-2 virus. And so, yeah, that's basically a sample of the graph that we've made. Here, this graph is quite small, but depending on the task that you want to have, you can play with much larger graphs. That's specifically relevant for downstream computational tasks. Okay, so now let's go to the last use case that I will discuss today. This use case, we have already discussed about it in the previous webinar. So if you want more details, I can refer you to that. Here, I will simply show you a little bit how we proceed to do that with the BioStrands module. So to give a little bit some context, basically, we have a subgraph that encompasses a relation between genes and concepts, and that we have extracted that from a PubMed abstract. And on top of this subgraph, we can start doing some analysis to perform enrichment, well, differential gene analysis enrichment, to understand a little bit more what you get back from RNAseq experiment. And so how to do that with the BioStrand module? First, you need to define the input file. So this is coming from RNAseq experiments. So ideally, what you want to have is a file that contains a gene count. So basically, all the reads that have been mapped to a corresponding gene need to be counted between different samples. And so here we define different samples. There are 6 samples in total, and these different reads have been mapped to genes. And instead of using the gene symbol here, we're using the ensemble IDs as a way to get these unambiguously annotate the data. And so this is done for all the genes that are found in our experiments. And we have also a file that is used for phenotype group. And this file format is exactly the same as what you need to feed to JANASOQ in addition to some other metadata. So basically, you define the gene count and you have a phenotype type that actually explicit what kind of experiment you are doing. So in this case, we have 3 groups that fall within the same phenotype and 3 other groups that fall within the same phenotype. And these 2 groups basically correspond to your test and case group from your experiment, on top of which you want to perform the differential gene expression analysis. And basically, I put these 2 files into the local input files repository, where and I have my notebook in the same repository that contains this sub repository. And once this is done, I can start doing my analysis. And for that, I simply need to import the corresponding client to perform this task. I simply initiate this client by providing the input file that I need in order to do this analysis. And here, there is a lot of things that is done in the background that I will not disclose in this talk. It's not really the topic, but let's just say that it will provide all the relevant statistics in order to build a graph that contains the enrichment information. So once this is done, we simply need to make the graph. And once the graph is made, we can simply open it, this time again with Hyfte, for example. And so basically what we are doing here, the kind of graph that we get from this application is a graph that looks like this, where for the size of the node have been associated to a p-value that assesses the statistical relevance of the concept with respect to what we have mapped previously. So it's very similar to a standard gene enrichment analysis in the way that we do this processing, but this time you have direct relationship between the different concepts appearing here. So you see this is actually a graph, and each edges between 2 nodes in the graph correspond to actual semantic relationships between these different entities. And with this, well, you can have a bottom and domain agnostic way to generate these enrichment analysis based on your experiment. And you can use that to discover novel pathways if it has not been found previously, explicit structure and so on, based on what you have in the literature. So this is all I had to say for the small use case. So I will leave now the talk to Arnout.
Thanks, Sébastien. Indeed. To conclude our webinar, we'll also have a quick session to handle some of your questions. So you can always leave them in the chat, and then we'll pick them up in a short while. But to conclude, I think with what we've been saying, especially in the beginning, we have a big fundament to make use of NLP tools in the broadest sense. So the perfect technology set out there to combine or utilize transformers or combination of transformers, dictionaries, and multiple ontologies, all from a notebook accessible, ready to play around with that, with your own code or your own Python skills on top of that. A nice way to, well, directly assess and integrate multiple domains, knowledge domains, information from multiple sources at hand to construct any type of analysis you want. But also very important, and it's something that we didn't highlight here, but is of course open to the full exploration once you have these tools at hand, is because we have so a nicely structured and deeply integrated relationship between sequences and text. Of course, well, that's the perfect starting point to exploit all this data using well, AI and machine learning tools on top of it. And well, in the end, this intrinsic connection between the omics, so your experiments, as Sébastien shows the RNAseq experiments, for example, and an immediate association and relation to what is out there in a certain knowledge domain. In this case, we checked it across PubMed. And it also brings us to the fact that, well, okay, we have all these pieces and bits of pieces of technology available, ready to use in any setting. And that's especially, I think, a very important part that it can help to accelerate certain drug discovery pipelines. And not all pipelines are exactly the same, but that's also the nice thing, that it is versatile. It can be used in any type of setting, implemented inside any existing pipeline workflow or notebook. What's so nice is that, well, it can aid in this, call it, data-driven hypothesis generation and validation. So whenever you have a list of concepts between, and you want to check associations between certain genes, diseases, interactions between antibodies, and so on, you immediately have the tools at hand to check in literature if anything is known around these couples or triplets in this case, or on a more extensive basis, if they co-occur or coexist in certain articles. That can greatly help you to plow through all the different articles in one go and have it in one overview. Next, the fact that you have this intrinsic couple between the sequence and the literature can also help you to, on the end of your pipeline, validate certain hypotheses which you laid out in your experiments. And well, all this to, again, accelerate the interpretation of your experiments, your thoughts, and build new hypotheses. Well, I think to quickly summarize it, so any starting point can, in your experiment, it can be an expression analysis or a certain association found within sequences like epitope-paratope twins, protein-protein interactions. Immediately from those associations, you can find all the relevant articles that can help you to decide whether there's a direct relationship between, for example, a gene and a certain mutation, or whether some pathways are known or described that are not immediately found in existing pathway databases, further information on modes of action, adverse effects, and so on. And this in its part helps you to interpret your data faster, but most importantly, it's data-driven. So it's association-based, knowledge-based, and this helps you to then further evaluate or redesign experiments based on what you find, and make better stop-go decisions, or identify new experiments and start a new round of experiments. A quick view on what we've planned in next steps, and probably we'll be talking about in a future webinar, is our ongoing developments regarding this entity recognition and named entity normalization steps to provide more control and have a better well, interaction with these items. Next to an extension towards well, new pre-integrated databases. So at this point, we can integrate any data source, but we are working to also have a default integration with pattern databases, for example, and open access full text. In the end, all we want to provide you with is a knowledge base queryable in a very efficient manner that can use any normalized entity and have all the relationships pre-identified as a perfect starting point. So I see some questions rolled in. So to quickly conclude our final 5 minutes, I'd like to see if I can direct some questions. Maybe I see a first one is one associated to the first part of Dirk is can you compensate the leftovers due to classical transformation usage? Something you can elaborate a little bit on, please.
Yeah, well, the first step is indeed identifying this semantic word boundary. So as if you know exactly, okay, these are the whole multi-word group, that's the first step you need to have. And then, of course, by looking at, okay, what is the mapping that you have from, for instance, from my transformer, you can see, okay, do I have an exact match, a full match with my discovered multi-word group, or is it only partial? And then you can further start analyzing, okay, even especially in like in English, on the it's words that are more on the right side of the concept are defining the let's say the meaning or the semantic class. So in that way you can further improve or your dictionary and see where the gaps are still in your predefined ontology or dictionary. Or another path that we are now actually working on is also further improve the transformer models as such, because again, we have the right chunks, we know what to analyze and what is the context around such meaningful chunks.
Well, to connect that to a different question I have here is regarding the use of dictionaries. Is it possible at this moment to combine your own dictionaries or ontologies in our system?
Yeah, and that's indeed it. So we are very flexible also to incorporate your own dictionaries. And again, like what I said just before, also immediately show where the gaps are in your dictionary. And so it can help you to improve that part of your own dictionaries and ontologies. Yeah.
Perfect. I see another question coming in. Yes, this webinar is recorded and soon after we conclude, you will be sent a recording. So that's taken care of. Yeah, one final question maybe before we conclude is, well, can you work with private databases? Can it also comprise of different languages?
Well, yeah, indeed. So the NLP part and the first step is indeed can be done in 11 different languages. So yeah, you have a whole range of possibilities there. And of course, you can also index your own data, so your own private data or data that's coming from electronic patient record systems and so on, as long as you have access and the rights to index data, we can do that.
Perfect. So I think that wraps up our webinar. I thank you all for listening. In case you have questions that you want to have addressed further, I sent you in the chat a link to which you can book a meeting. If you have a question you want to have addressed in a more private setting, I welcome you to use that link. If you have further questions, don't hesitate to contact us. And I'd like to see you again in our future webinar.